Yeah, so I'm going to talk about, one of my favorite topics, which is omega-3 fatty acids and their role in controlling inflammation. So inflammation is a physiological process. We can measure the inflammatory response, for example, by measuring activated cells in people's blood, measuring the concentration of cytokines in people's blood, measuring acute phase proteins like C-reactive protein in people's blood. So we can measure the response. That response is initiated by exposure to triggers. Most often, we think of microbial components like lipopolysaccharide as the trigger for inflammation. But other triggers can include tissue damage, metabolic stress, and oxidative stress. So the link between the trigger and the response is the pathways that are initiated upon exposure to the trigger. That's because cells involved in inflammation have receptors that recognize, for example, microbial components or damaged tissue. These receptors initiate signaling processes that cause cellular activation and the upregulation of genes that code for inflammatory proteins. So there's a link between the
Uh,
triggers.
Please, Professor Calder, your slides is not moving.
Oh, okay. Okay, I'm on the. Okay, I probably have to come out of my presentation then. So, okay, so you can see my presentation.
Okay.
Yep. Okay, I put it back in presentation mode. And can, can you see the second slide now?
Not yet.
No, we can see your full screen presentation. I think you should select your full screen.
Yeah, I have done that. Yes. So how do you see it now?
Yeah, it is moving.
Okay, I'm gonna put it on presentation mode. And I go to the second slide. Do you see that?
No, it is
No.
it isn't moving.
Okay.
You don't need to full screen. You can just click it and.
Okay, so you can see it now, not on full screen.
No, it's not full screen, but
Okay.
now
Yeah.
slides are moving. So
Why don't you show us
Yes.
slide by slide? Slides.
Yep, sure. Thank you. Yeah. So I was talking about the response, which can be measured by activated cells, cytokines, acute phase proteins. I mentioned the triggers, microbial components, and damaged tissue, for example, metabolic stress, and the link between the trigger and the response being the pathways that are initiated through receptors recognizing the triggers and generating inflammatory signaling. So the 5 cardinal signs of inflammation are heat, redness, swelling, pain, and loss of function. These signs are caused by the chemicals that are produced as part of the inflammatory response. As I mentioned, inflammation is a physiological response, and we now know that it has 2 phases, the initiation and propagation phase and the resolution phase. In other words, inflammation is triggered, and when it's run its course physiologically, the resolution phase begins and the system returns to the baseline level of inflammation. So if we put all of that together, physiological inflammation involves initiation, propagation, and resolution. So it's self-limiting. The question is, what happens if there's no resolution? So on the left, I show you what I've just shown you, which is the self-limiting physiological inflammation. And on the right, I present to you what happens if the resolution phase is not working well. So we get continued chronic inflammation with metabolic changes, tissue damage, pathology, loss of function, and even death. So for example, the cytokine storm in COVID-19, which was responsible for most of the mortality, was due to loss of resolution of the inflammatory response. So this is really high-grade inflammation where we can measure extremely high levels of CRP, for example, in the blood of the patient. But we also now recognize there's a condition of low-grade inflammation with rather modest elevation in CRP concentration. So we can call this silent inflammation, if you like. So the presence of the chronic inflammation is not well recognized. So we can have a high-grade inflammatory response, we can have a low-grade inflammatory response. Both of these are associated with different conditions and diseases. So on the left, I show those diseases that involve high-grade inflammation, things like arthritis, inflammatory bowel disease, asthma, multiple sclerosis, cancer, cachexia, and sepsis. On the right, I show the conditions that involve low-grade inflammation, cardiovascular disease, metabolic disease, cognitive decline, many cancers, loss of lean mass, and immune decline. Many of these non-communicable diseases are associated with aging, and the elevation of low-grade inflammation as we age is termed inflammaging. So we think inflammaging is part of the risk factor profile with these common non-communicable diseases. So my point is that inflammation can be associated with high-grade chronic inflammatory diseases, but is also part of the risk factor profile for these low-grade non-communicable diseases. So what's the link with fatty acids? So the key link, at least initially, is the recognition that the omega-6 fatty acid, arachidonic acid, is a precursor of important lipid mediators called oxylipins that are involved in inflammation. These include the prostaglandins and leukotrienes. And of course, there are many anti-inflammatory pharmaceuticals that target arachidonic acid metabolism, including aspirin and other non-steroidal anti-inflammatory drugs, and specific enzyme inhibitors like the COX-2 inhibitors. So mediators produced from this omega-6 fatty acid are intimately involved in inflammation. They act as mediators and regulators of the inflammatory response. So there's a view that too much arachidonic acid or an excess of arachidonic acid metabolism sets the environment up to be a pro-inflammatory environment. And this is where the omega-3 fatty acids become very important. So I'm going to talk about two biologically active omega-3 fatty acids, EPA or eicosapentaenoic acid and DHA, docosahexaenoic acid. These are very long chain, highly unsaturated fatty acids. When we increase the intake of these fatty acids, they're very easily incorporated into our cell membranes. So this is an old study we did where we measured the amounts of polyunsaturated fatty acids in the phospholipids of neutrophils. So this is the membrane of these cells involved in inflammation after 12 weeks of supplementation. And we supplemented people with increasing doses of tuna oil, which is a DHA-rich oil. So we found the higher the dose of tuna oil, the greater the amount of EPA and DHA that appeared in the membrane of these cells. But as EPA and DHA are incorporated, so the amount of arachidonic acid is decreased. So we diminish the amount of this important precursor for pro-inflammatory lipid mediators, and we increase the amounts of EPA and DHA that are present. The effect of this change in fatty acid composition of the cells is shown here in data from another one of our studies, which was a dose-response study in humans with increasing amounts of eicosapentaenoic acid given every day for 3 months. We took blood cells, white blood cells from the people before the study, that's the 4 red bars, and after the study, that's the 4 yellow bars, and we exposed the white cells in a test tube to bacterial lipopolysaccharide, which induces an inflammatory response. And we measured prostaglandin E2 production. So you see in the control group, there's no change, which is what you would expect. In the low dose EPA group, there's also no change. But as we increase EPA intake, we get a significant dose-dependent reduction in the production of this mediator from arachidonic acid. So this is just an example of those sorts of studies which have been repeated many times in vitro, in animal experiments, and in human studies. So EPA and DHA prevent so much arachidonic acid getting into inflammatory cell membranes, and they interfere with arachidonic acid metabolism, so decreasing the production of these pro-inflammatory eicosanoid mediators like prostaglandin E2, prostaglandin D2, and the 4-series leukotrienes. But of course, inflammation is about more than lipid mediators. It involves many protein mediators like cytokines and chemokines. And cytokines like tumor necrosis factor, interleukin-1 and interleukin-6 are involved in all of the inflammatory states, both the high-grade inflammatory states and the low-grade inflammatory states that I mentioned before. So again, in animal feeding studies this time, we and others have shown that feeding omega-3 fatty acids in the form of fish oil decreases the ability of inflammatory cells to produce tumor necrosis factor. So this is a mouse feeding study where 3 diets were fed, then macrophages were taken from the animals, and they were either not stimulated, that's the purple bars, or they were stimulated with bacterial lipopolysaccharide, that's the green bars. So firstly, you see stimulation increases expression of the tumor necrosis factor gene and increases TNF protein production. But if the cells come from animals fed fish oil, there's a much reduced production of TNF and much reduced TNF gene expression. We found similar results for the other classic inflammatory cytokines, interleukin-1 and interleukin-6, where omega-3 fatty acid feeding decreases the ability of cells to produce those inflammatory cytokines when they're stimulated with lipopolysaccharide. And actually, the anti-inflammatory cytokine interleukin-10 is increased with omega-3 fatty acid feeding. Now, I won't go into the mechanism of action by which EPA and DHA interfere with the production of inflammatory cytokines and chemokines, but it involves a downregulation of the transcription factor, nuclear factor kappa B, so that there's less translocation of active NF kappa B to the nucleus. So there's actually interference in NF kappa B signaling by omega-3 fatty acids. And we now know that this effect actually is mediated at the level of the cell membrane. So when the cells receive the inflammatory stimulus, so they're exposed to an inflammatory trigger, there's movement of proteins within the cell membrane to form signaling platforms called rafts. And we now know that when the membrane is enriched in EPA and DHA, the ability to produce or to assemble those rafts is impaired. So the downregulation of NF kappa B signaling with EPA and DHA is actually due to an impairment in raft assembly at the membrane level. So omega-3 fatty acids are acting to decrease the protein mediators of inflammation and the lipid mediators of inflammation through the mechanisms that I've alluded to. I just want to go back now to inflammation resolution and the importance of resolution of inflammation in protecting the host against the adverse impacts of the inflammatory response. So researchers looked in model systems at what chemicals were produced during the resolution phase of inflammation, because this is actually an active process. And they identified new lipid mediators formed during the resolution of inflammation. And according to their structure, they termed them resolvins because they resolved inflammation, protectins because they protect against inflammation, and merisins because they're macrophage-derived resolving mediators. Together, resolvins, protectins, and merisins are termed specialized pro-resolution mediators, or SPMs. Now, the really exciting aspect of this new biology of resolution of inflammation is that these specialized pro-resolution mediators, the SPMs, are produced from EPA and DHA. So those pathways are outlined here. So EPA gives rise to E-series resolvins, DHA gives rise to D-series resolvins, protectins, and merisins. So specialized pro-resolving mediators are produced from omega-3 fatty acids. So the idea is the trigger induces acute inflammation, and physiologically that resolves, and part of the resolution pathway involves the production of omega-3 derived specialized pro-resolving mediators. And this resolves the inflammation and allows tissue repair, healing, and return to homeostasis. If the acute inflammation doesn't resolve, it becomes chronic. And that goes on to cause the deleterious consequences of inflammation, like tissue damage, persistent infection, autoimmunity, and so on. So in model systems, it's been demonstrated that providing the omega-3 derived SPMs rescues chronic inflammation and drives resolution. So one argument about loss of resolution is insufficient supply of omega-3 fatty acids to support the resolution process. So that's an interesting new angle on inflammation. So what I've added to my mechanistic scheme here is the production of specialized pro-resolving mediators leading to resolution of inflammation. So omega-3 fatty acids are both anti-inflammatory, so they decrease inflammation, but they also give rise to the chemicals that resolve inflammation. So they're anti-inflammatory and pro-resolving. So what I've talked about so far is really findings on mechanisms from animal models, and also I showed you data on incorporation of omega-3s and prostaglandin E2 production from human studies. So the question is, do omega-3s actually have anti-inflammatory actions in humans? So there are now dozens of studies where omega-3 supplements have been used, and inflammatory chemicals have been measured in the blood of older people, people with inflammatory conditions, and so on. And these data from human trials have been aggregated in many meta-analyses. And I just pick out some here showing that omega-3s decrease inflammation in people with diabetes, with MS, and here in mixed participants. So in many conditions. So just to summarize now, inflammation is part of host defense, but it can be damaging if it's not controlled. And this links inflammation with many human diseases. I've talked about how the omega-3s, EPA and DHA, act through several mechanisms to modulate inflammation, and these mechanisms link the cell membrane with the nucleus. Resolution of inflammation is driven by omega-3s via specialized pro-resolving mediators. Human studies confirm that EPA and DHA reduce inflammation, and I think the anti-inflammatory and pro-resolving actions of EPA and DHA are relevant to the prevention and treatment of conditions with an inflammatory component, and these include the common age-related conditions. And for this reason, I consider EPA and DHA to be the master controllers of inflammation. So thanks very much for listening, and thanks for having me at your meeting today. So I'll stop sharing now.
Okay, thank you so much, Professor Calder, for that enlightening presentation on the mechanisms of inflammation and the powerful role of omega-3 fatty acids play in control inflammation. And specialized pro-resolving mediators were particularly impressive and insightful. Now let's take one or two brief questions before we move on. And after all the presentation, we will continue with a more in-depth panel discussion. I'd like to invite our panelist, Professor Myung-ho Kim, to share his thought or pose any question at this time.
Yes, thanks for your presentation. It was really helpful to deeply understand how polyunsaturated fatty acids have anti-inflammatory effect. So, as a clinician, I see the cancer patient. So I'm interested in managing low-grade inflammation in cancer patient. So I was wondering if there is any report or evidence of supplementation of polyunsaturated fatty acid, their anti-inflammatory effect in cancer patient, and also is there any evidence of improving cancer patient prognosis, such as survivor or disease-free survivor
Yeah, yeah, sure.
mortality?
Yeah, that's a great question. So of course there's some research in this area, and I'm going to talk about three different aspects. So one is in prevention. So there are some data, mainly from epidemiological studies, that some cancers, the risk can be reduced if people are using or eating more omega-3s over a long period of time. And this might be about creating a less inflammatory environment, of course. So I think they have a role in prevention of some cancers for sure. The second is in cancer surgery, particularly for gastrointestinal cancer, where there is quite a lot of evidence that using omega-3s perioperatively improves outcomes like infectious complications and length of stay. But you're asking about the middle ground, which is if you have a cancer patient who's maybe receiving, radio or chemotherapy, for example.
Yeah.
And there, of
During
course, there's a
chemotherapy,
problem. Yeah.
during therapy or after chemotherapy in the surveillance period.
Yeah. So I think inflammation, of course, is a big problem for those patients. And I think it's probably somewhere between low grade and high grade, actually. So I think there could be a role. But I'm not, if I'm honest, I'm not familiar with the literature in that part of the cancer spectrum. But I think there will be a role in terms of controlling the inflammation, which is actually one of the issues that those patients can have. So certainly it would be worth looking into the literature in that area.
Yeah, thanks for your comment. So the reason why I am interested in the low-grade inflammation in cancer patients is that the cancer patient, the tumor cancer itself can induce inflammation, and also the patient with cancer cachexia, they also will have low-grade inflammation. So I think managing that low-grade inflammation is important to taking care of the patient.
Yeah, you're right. So I've been thinking a little bit more while you've been talking. So there are a couple of studies from Canadian researchers who have used omega-3s during I think chemotherapy, and what they found was there was less toxicity and more cycles of therapy can be used. And there's also a big interest, as you mentioned, on prevention of loss of muscle mass. So the induction of sarcopenia and cachexia. So there are studies looking at that. I think it is a fruitful area for sure.
Thank you.
Okay.
Last question.